Okay. You showed how adjusting system sensitivity by increasing or reducing receptor expression can reveal hidden properties of partial agonism, panagonism, and inverse agonism. So, in real world drug discovery, how can researchers use this concept of assay volume control to avoid misclassifying compounds early on? For example, mistaking a low efficacy agonist for a silent antagonist. Yeah, excellent. That's spot on. So, first of all, I really highly support having at least a low sensitivity and a high sensitivity functional assay in your program, just to take care of this, to be prepared for. Because when you have that, you can quantify. That doesn't mean you'll see it in vivo, but it'll certainly prepare you. There's a lot of examples where unprepared programs got a nasty surprise, especially with agonism, positive agonism. When prenolterol was developed in Sweden, they used systems that generally were not that highly coupled, and so the tachycardia, which you do not want in heart failure, really was very low and didn't seem to be important. And then when they put it in humans and other systems, more sensitive, they got a nasty surprise. They got their heart rate going up when they don't want that. So being prepared for it. How do you know? Well, you usually will use a benchmark. For example, let's return to the beta receptor example. There's a nice range of partial agonists: pyrbuterol, prenolterol, pimidolol, that you can use and sort of as a marker, and you can say, well, in humans, this stuff just starts to raise heart rate. That'll be your benchmark. And then in vitro, compare all your agonists to that one. And anything higher, you can say, that'll probably cause heart rate increases. Anything lower, that probably won't. So that's how you translate these differences in volume control. I'll add one last thing. Inverse agonism, you detect only in a constitutive system. As a general rule, almost all antagonists are inverse agonists, but it's rarely relevant. It's when it was discovered, there was great excitement. Oh, here's a new therapeutic tool. Very few natural systems are constitutively active. Nature doesn't like that, I think. Doesn't want systems running rampant. They want hormonal and transmitter control. The few systems like ghrelin, which is a constitutively active system with appetite, that one may be physiological, but by and large, your inverse agonism will not be a therapeutic property. So why look for it? Because it can be linked to a negative thing. It has been associated with antagonist tolerance. With chronic use, you will get increased receptor number and escape from the therapeutic effect. That has been the case where it was first seen were the H2 inhibitors for ulcer, where the inverse agonists showed tolerance and the neutrals did not. Will this always happen with every inverse? No, because it depends on the cell type, how quickly receptors are made, transported, internalized, and the receptor type. But it's a property you should probably know about for your antagonist and say, okay, this one's an inverse. Let's keep our eyes open for possible tolerance in future. But just having this volume control greatly expands your program to expect, well, to understand what you may not expect.
And just to add to that, Terry, this is where the importance of having a low expression and a high expression cell line in which you can test your compounds that'll allow you to tease out these properties.
Absolutely right. Absolutely right. Yep.